Hello viewers, welcome to my channel ABC. Today I am here to discuss another new biological topic which is very important and it is very common in all the classes including all boards. It is for class 10, class 11 as well as for class 12. It's similarly important. Only some detail will be less or more, but everything will be common in our understanding. So what is gene? This is the topic. Gene, where gene is resides. We know a unit of heredity which is transferred from a parent to offspring, means from parent means father and mother to their child, and is held to determine some characteristics of the offspring. Offspring having some character which are derived from their parents. So we can see here the genes actually resides inside any cell nucleus where chromatin fibers are found. It is a thread-like structure. Inside this, some chromosomes are there. These chromosomes having some DNA inside it, and inside DNA there will be some gene. So if we see inside a chromosome, then we can see a chromosome having two chromatids. This one, another one. These two chromatids are actually held together by a centromere in central region. It may be in lower or upper side also. But when we unwing these wrapped up fibers, we can see chromatin fibers like this. And inside these chromatin fibers, we can see some dots like beads on string. So these are the beads. These are actually called nucleosomes. These are having some protein inside that we will discuss just now. And when we more unwrapping the fibers, we can see DNA double helix inside these nucleosome structures. And after DNA, we can get genes on the DNAs. That is some dot points like this. These are genes. Now, if we see more about nucleosomes, then we can see nucleosome is made up of two types of things. One is protein and another is DNA. The proteins are actually wrapped up by the DNA. Finally, we can see some ball-like structure which is actually a 3D structure. You can see here a cube. Inside a cube, there will be some balls. 8 balls actually present inside. These 8 balls are actually H2A, H2B, H3 and H4, each of 2. That means each kind will be present twice. So finally, there will be 8 number of balls present for histone proteins. These are called core histone protein. It will actually look from outside like this. You can see a ball like this. Here, 1, 2, 3, 4. Behind this 5, behind this 6, behind this 7 and behind this 8. Finally, from outside you can see these are H2A, then it is 2B, this is H3 and this is H4. Each of 2 types, that means twice. H2A will be twice, 2B will be twice, H3 will be twice and H4 will be twice. Finally, it will be wrapped up by a DNA thread. A DNA thread will wrap it first and second and approx half time. And outside this ball, that means core histone. This is called core histone octamer because it is present in center. And outside this ball, there will be 2 balls. Here one H1, another one is H1 here. Finally, we can see DNA, which is wrapped histone or core histone, due to DNA having negative charge and core histone or histone protein having positive charge. So they can attract each other. So they are always wrapped up. Now we can see this final structure having H1 protein outside core histone, H2A twice, H2B twice, H3 twice and H4 twice. Ultimately 8 balls present inside the core histone. So it is called histone octamer. What is DNA? deoxyribonucleic acid. In 1953, DNA double helix model was given by actually 4 scientists togetherly worked on this research. But finally, we came to know that in 1953, DNA double helix model was given by Watson and Crick. But for all research, etc., Nobel Prize was given to 3 scientists: James Watson, full name, Francis Crick, with Maurice Wilkins, though another scientist was there in the total team, named E. Rosalind Franklin. Actually, she expired earlier in 1958, so did not get that award. Now, we are getting DNA structure. DNA structure, if you see, there are 3 main things. One is pentose sugar, it's 5 carbonous sugar, nitrogenous bases, and phosphoric acid. Pentose sugar is actually deoxyribose in that case, because another thing is there, we know that RNA, the single helix, that is ribonucleic acid is there, but here it is deoxyribose sugar. We can see the structure, that 1, 2, 3, 4 and 5 carbons here, and mostly the 3 and 5, these 2 carbons are the attachment point for any strand that we will see later. Now, nitrogenous bases, mainly 2 types, one is purine, another is pyrimidines or pyrimidine. Purine, adenine, guanine, and pyrimidine is cytosine and thymine. In case of RNA only, it is uracil. In case of RNA, it is uracil instead of thymine. So we can see here, that is adenine. Adenine having always double bond, and guanine having always triple bond, and cytosine having always triple bond, thymine or uracil, whatever it is, of double bond. So whenever there is a bonding between 2 things, purine and pyrimidine, because they are complementary to each other. Purine and pyrimidine are complementary to each other. Without purine, pyrimidine cannot work, or without pyrimidine, purine cannot work. Actually, adenine is always linked to thymine, and cytosine is always linked to guanine. This is the sequence due to their bond matching. And one thing we should remember more, that adenine and guanine having a structure of double ring. One is hexagon, another is pentagon. Hexagon is called pyrimidine ring, which is similar to, in case of pyrimidines, thymine, cytosine or uracil, having single hexagon ring. And with this, in case of purine, we can see with a purine another pentose, that means 5 pentagonous ring is called pyrrole ring. It is called pyrrole ring. It is lacking in case of pyrimidines. So we have come to know that another thing is phosphoric acid or phosphate, that link them in a particular sequence. We'll see the sequence now. Generally, I used to draw the drawing in between times to save some time. Now we can see the double helix structure of DNA given by Watson and Crick. Here we can see 2 anti-parallel strands. One is starting from 5' and going towards down to 3'. Another is from 5' again going towards up to 3'. This prime is nothing but the positions of carbon in pentose sugar. We have seen it earlier. 1, 2, 3 and 4. Here it is 4 and 5. The 5 carbons here. And if we link something in 3 position or 5 position, it will see like this: 3' and 5', and in anti-parallel strand again 5' and 3'. So we can see one helix, one complete helix for DNA here from this much, and we can have the size 34 angstrom, and the breadth or width it is 20 angstrom. Though we know 6 types of DNA yet found, that is called A DNA, B DNA, C DNA, D DNA, E DNA and Z DNA. In each type A to E, we have right-handed structure, and one only the Z DNA having the left-handed structure. And we are drawing here the B DNA, that is the B DNA, structure of B DNA. It is 34 angstroms mostly found, so we are drawing it. and 20 angstrom in thickness. Now we can get in one total helix, there are 1, 2, 3, 4 and 5. Again 6, 7, 8, 9 and 10. That means in total one helix we can see 10 strands. These 10 strands are situated in equal distance, so we can get 3.4 angstrom for the minimum gap of 2 strands. That is universal in case of BDNA. Now we can see more that there will be these strands are made up of mainly adenine, then it will be linked to thymine, and cytosine, then it will be linked to guanine. As per our previous discussion, we know purine and pyrimidine are complementary to each other and they are linked with hydrogen bonds. Here it is adenine and thymine having double bond in each case, so they are attached with 2 hydrogen bonds here. And in case of cytosine and guanine, we can see 3 bonds. So as per Chargaff's rule says that due to complementarity of the 2 base pairs, purine and pyrimidine will always be in same quantity in a particular type of DNA, and adenine and thymine will be equal, and cytosine and guanine also will be equal. So we can see here more structural content, that here it is a small groove here, so it is called minor groove. It is called minor groove, and here it is again a larger groove. We can better call it major groove, or here we can see again the major groove, and again here it is a minor groove. So if this one is minor groove, here we can draw here also it is major groove. We have seen here in DNA, in each strand there is a complementarity rule as per Chargaff's rule, and we have concluded here A always be with the T equal and C always equal with G as in pairs. So the rule becomes A plus G equal to C plus T, and therefore we can write A plus G by C plus T is equal to 1. That means the ratio is 1:1 in each case. 1:1 in each case. Now we can see more in detail. In each strand there are nitrogenous bases, and for each 3 nitrogenous bases we can see one gene, that is called one gene. Suppose in case of DNA we have here T, A, C. That means these 3 makes a gene. Here in this opposite side then it should be G, T and A. That is also a gene, and this way numerous genes are arranged in the DNA. In case of human beings, we have said at least the 2 point something meter, that means more than 2 meter length of a DNA we can find in humans. We can see here in more detail that there is a central line in between of a DNA, which you can see here that a particular line is drawn here as midline, where left side and right side containing different types of nitrogenous bases, and they are bonded with hydrogen bonds, where some double bond or triple bond may be there, but the bonds will qualify as nucleoside. Here how they form nucleoside? We can see nucleoside is formed of any nitrogen base when added with pentose sugar, it will form nucleoside. Side means one side. Here we can see this side and again this side. These two are complementary, but these are different sides. Now when we add phosphate group as phosphoric acid with any type of nucleoside, it will form nucleotide. So nucleoside plus phosphate group will produce nucleotide. We can better conclude here, nucleoside plus phosphoric acid will form nucleotide. nucleotide will form like this. Here the example we can see, when adenine is added with any pentose sugar, it will form deoxyadenosine as nucleoside. When guanine is added with any pentose sugar, it will form deoxyguanosine. When cytosine is added with any type of pentose sugar, it will form deoxycytidine. And when thymine is added with any pentose sugar, it will form deoxythymidine. Such a way, when deoxyadenosine is added with phosphate, any type of phosphate as phosphoric acid, it will form deoxyadenylic acid as nucleotide. Again, if it added guanosine with any phosphate as phosphoric acid, we can see here, we can better write like this. Now guanosine, it will form deoxyguanylic acid. Then we can see cytidine, and deoxycytidine, it will form with phosphate deoxycytidylic acid, and for thymine it will form deoxythymidylic acid. So deoxyadenosine is nucleoside, so deoxyadenylic acid is nucleotide. This is the difference between nucleoside and nucleotide. It is very important, it comes in exam. What is the difference between nucleoside and nucleotide? Now we have to see something more about DNA. Regarding DNA replication, we know here there is a replication fork, and this system of replication comes from the replication different theories like conservative theories, dispersive theory, another is semi-conservative theory by Watson and Crick. So now we have seen here, due to helicase enzyme, this particular DNA double helix strand will just open. That means the hydrogen bond in between is broken by this helicase enzyme. So two different forks are formed, and ultimately one strand is from 5' to 3' is a continuous strand. Another is discontinuous strand which is anti-parallel, that means 5' to 3'. And there, when new strand is synthesized, then this side forming a continuous new strand which is called leading strand, and another one from this strand, from upside to downside, there are some small pieces of small strands are generated. This 5' to 3' each, and these strands are called actually lagging strand. And due to this fragmentation, this is called Okazaki fragment. This is called Okazaki fragments. These are later added by enzyme ligase. We know here helicase enzyme functions, and when the complete two set of DNA is formed, the newly formed DNA and the old one looks like this, and the new one in red color I can show you. So two new strands formed from the previous one DNA. That new formed DNA is formed due to 50% previous means old DNA and 50% new DNA. 50% old and 50% new DNA is synthesized here. So ultimately that new DNA produced from semi-conservative theory. That means the theory proves that 50% new DNA is there and 50% old DNA is also there. So it is called semi-conservative theory by Watson and Crick. Now I have reached to the end of this today's discussion, and about the chromosome DNA, all the topics we have covered, and next very soon we will come with a new topic of these chapters. And if you understood the topic, today's topic, then please like my video and share this video for your friends' benefit, and must subscribe my channel very soon. Hit the bell icon for further notification. If you have any type of queries or questions or any suggestions, please put it on the comment box. And stay at home for the lockdown period. Also, study from home carefully. Be healthy and safe. Thank you.